are Gene Online, your premier biotech reporting agency. Today, we have the pleasure of speaking with Dr. Alex Huang, Vice President and Head of Cell Therapy at BeiGene. Dr. Huang, welcome.
Thank you very much.
To kick things off today, we're very interested in understanding more about BeiGene's focus on iPSC derived cells. Could you tell us why BeiGene cell therapy technology is focused on iPSC derived cells, how this approach compares to other allogenic cell therapies, and what the potential benefits of using iPSC derived cells are?
The uniqueness about this platform is that the entire therapy can derive from one single clone that harbors all the genetic modifications that you like. And then you differentiate it and expand it with a very uniform methodology. So you can ensure the homogeneous population, cell therapy population. Now, this actually is quite different in terms of the variation that the autologous field and the donor-based allogeneic cell therapy field they face. Now, the second point also is that since cell is the therapeutics in the cell therapy, so you want to make your cells as potent as possible. And sometimes that will require multiple gene edits. In the context of multiple gene edits, it's very difficult for autologous or the donor-based derived or donor-based allogeneic cell therapy to derive a homogeneous therapeutic population. But that's not a problem for iPSC, as I mentioned earlier. So I'm very excited about this. I really think that if we get the iPSC-derived cell therapy to work, it could change the entire cell therapy landscape.
BeiGene has recently achieved an incredible milestone by submitting 4 patent applications within just 22 months. What would you say are the core advantages driving this impressive achievement?
In order for the iPSC-derived cell therapy to be successful, number one, I think that one has to have a very well-characterized GMP-grade iPSC clone. And that clone has to have a very efficient differentiation and expansion capability. The second is that on this iPSC, you have to derive a proprietary genetic alteration that can ultimately enhance the efficacy and the longevity of your product. The third thing, and the most important thing, is that you gotta have a proprietary differentiation and expansion methodology. And that has to be in a feeder-free format in order to lower the manufacturing risk. And I think we have all these three, and those are what the patents are really based on.
But transitioning to a bit of a broader perspective, as the head of cell therapy at BeiGene, how do you see BeiGene positioning itself specifically in the field of cell therapy over the next 5 years?
BeiGene as a company, we put a lot of emphasis and focus on delivering affordable and accessible medicine, not just in the biologics, not just in small molecule, but we want to do it for the cellular medicines as well. So from this iPSC-derived cell therapy platform, what we're hoping in the next 5 years, we'll be able to derive to a proof of concept. And that would open up the floodgate of using the iPSC as the main go-to approach for the cell therapy. And that would fundamentally change the entire therapeutic landscape of the cell therapy and make it the most important thing for patients as accessible, as affordable as possible.
Wow, thank you so much, Dr. Huang. This has been incredibly insightful, and we look forward to seeing much more of BeiGene's continued success and contributions in the field of cell therapy.
Thank you so much. Thank you. Thank you for having me.